More than 35 years ago, shall we say. I was a postdoc in Germany. Don was working at the University of Wisconsin in the lab with Jerry Dempsey. He went on to the University of Northwestern University, where he did a postdoc with Jack Feldman. He got his own now. And then when Jack Feldman decided UCLA was more in his future, there was a position left behind, and there was somebody right ready to fill it. And Don's done a lot of work over the years on respiratory neuroanatomy, so the circuitry of the pulmonary stress receptors, broader chemoreceptors. I don't think anybody has presented that better. And he's developed new interest in the effects of metabolic hormones like leptin and the way that it influences some of those structures. So I'm going to leave it to Don to tell you the story the way you would like. We should have had you early in the semester. But it's very nice to be here. I appreciate the invitation. Thank you, Gordon.
So I want to make this as informal as possible. I've got potentially a lot of slides, but it would probably be more fun if I didn't get through all the slides. And so feel free to interject at any point. So you can disagree with me, that's fine. But discussion is always better than a passive audience. So to start off with, what are we talking about for respiratory control and neuroanatomy? So you've already had a number of lectures and covered pretty much everything that's on this first slide. So let me just go over it quickly. And what we're talking about is a brain that generates a basic respiratory rhythm that goes out to both the cranial motor neurons, control airway resistance, and the pump muscles, generating movement of the thorax and air movement in and out. And all that has to integrate very nicely and seamlessly, as we just heard from Paul, with behavior and other interoceptive kinds of things that are going on. And you heard from Jack Feldman, I'm sure, in great detail about intrinsic oscillation and the respiratory circuits in the brainstem and the pre-Betzinger complex and the parafacial respiratory group, most in particular, and from Gene Natty on things like central chemoreception that certainly adjust respiratory pattern. So I'm going to, in some ways, I think, provide a little bit of a review and hopefully point out some new things to you as we go along. So I'm going to start off by just going over the basics. And this is about as basic as it gets. It's just a recording from a phrenic nerve and abdominal EMG taken simultaneously from, in this case, from a decerebrate rat without vagi, and an increasing drive that you can see on the abdominal musculature in particular, with very little change in the phrenic. So all this by way of saying, you know, it's not just a massive output of respiratory drive that goes to all of the nerves and all of the respiratory muscles. It's something that has to be tailored. And if you look at the details of that motor output pattern on the abdominal EMG, you can see that it certainly shuts off during inspiration, but early on at low drives, there's not much early expiratory activity. As you get more and more drive, you get this initial burst of post-inspiratory activity that then declines through the initial part of expiration, followed by a large augmentation. So ideally, we'd like to explain how all this happens, how we generate this differential motor output. We've got a long ways to go to get there, but we do have some basics. So let's go over what we do know about. And first off, if we're going to talk about respiratory neuroanatomy, we sort of have to know what we're talking about. And this goes back historically, as long as people have looked at sort of central respiratory control. It's been defined based on the location of neurons that fire phasically, basically have an on-off pattern. Either they fire during inspiration and are silent during expiration, or vice versa. There are many shades on that, so there's a fairly extensive variety of subtleties that you can come up with on that. But all of these groups that we're going to talk about have some form of phasic respiratory activity, and hence that's how they were initially identified. And when you do that, you come up with, historically again, what have been three groups of respiratory neurons in the brainstem. So this is the pons and the medulla. And so Dorsally in the pons, or in the medulla rather, dorsomedially, you have what's referred to as the dorsal respiratory group, and it's associated with the nucleus of the solitary tract, which is that site of termination of primary afferents coming from the lungs and the airways and the vagus and the glossopharyngeal nerves. And then ventrolaterally in the medulla, you have that long column that's subcompartmentalized into a number of respiratory groups, but together it's referred to as the ventral respiratory column. And then separately from that is a group of neurons that's located rostro-dorsolaterally in the pons, referred to as the pontine respiratory group. It's a much more enigmatic group. Its function is, well, a lot of different functions have been ascribed to it, but none have been really rigorously demonstrated to be, this is the role of the pons. So, but let's go a little bit through some of these. So this, looking at the neuroanatomy, first off, of the lateral respiratory groups. So this would be including the ventral respiratory column and the pontine respiratory group. And so how do we do this? So what is done here is a retrograde tracer, fluorogold to be precise, is injected into the ventral respiratory group, rostral ventral respiratory group, a component of the ventral respiratory column, and you'll look to see where neurons are located in the brainstem to project there. And what you do when you do that, you look at a parasagittal section here, and you see now that neurons project sort of throughout that entire lateral aspect at some level. So looking rostrally, the rostral edge of the pons is up here. This is the transition to the spinal cord. There's the ventral, obviously, dorsal, cerebellum's up here. Few landmarks, this would be the facial nucleus, the lateral reticular nucleus, motor trigeminal. And you can see those on the right here. This is just a cholinesterase stain that shows up the motor neurons in the motor trigeminal, the facial nucleus, the compact division of the nucleus ambiguus, and then pedunculopontine neurons that are cholinergic, not motor neurons. And the pons, so even, you know, for most people, just differentiating between the pons and the medulla has been a challenge. And sort of if you go to this caudal edge of the brachium going into the cerebellum and draw a fairly direct line down to this incision that goes on the ventral surface of the medulla here, or at the transition, you get a nice straight line that includes then most of the facial nucleus in the medulla of a rat. If you do the same kind of analysis in a human, it's mostly in the pons. But you can sort of demark this region fairly well. And if you look at this retrogradely labeled population of neurons, what you do is see that there's not a ventral respiratory column and a pontine respiratory group, but there's more or less a continuous column of neurons that extends all the way from the cervical spinal cord on up to the rostral dorsolateral pons. It's interrupted a little bit by the facial nucleus, but if you look carefully here, you can see that indeed the retrotrapezoid nucleus, as it's referred to, and was talked about, I'm sure, in detail by Jack Feldman and Jean Natty, and the parafacial respiratory group, which is that group that Jack ascribes expiratory rhythm generation to. And there's even a fair number of neurons that are populated just dorsal to that facial nucleus, but nobody's really identified their particular functional role. And then this on the bottom right is just a missile section which just shows the cell groups. So the facial nucleus, motor trigeminal. A marker, if you're ever doing these sections and you're interested in identifying the right medial-lateral level to hit the parafacial and pre-Betzinger region and the semi-compact, compact divisions of nucleus ambiguous, the facial nerve, it goes up, remember it goes up, starts at the facial nucleus, goes up dorsally, and then reverses itself and comes back down ventrally and exits the pretty ventrally on the brainstem, and where it reaches on its downward path now in a parasagittal section, where it reaches just sort of the dorsal level of the facial nucleus is about the right level to find pre-Betzinger or Betzinger complexes in particular. All right. So what about rhythm and pattern generation and how they break down in these groups? So this is just same section showing the retrograde labeling and the model below and the various compartments. And so this is the ventral respiratory column. You'll see people refer to it slightly differently as to whether they include the retrotrapezoid and parafacial groups, which are just ventral to the facial nucleus, on back through the Betzinger, pre-Betzinger, rostral VRG, and caudal VRG, standing for the ventral respiratory group. And of those now, you find that rhythmogenesis is largely ascribed to the pre-Betzinger complex and the parafacial respiratory group. And so you have this group here with a Betzinger complex sort of sandwiched in between that is fundamentally involved in respiratory rhythm generation, both inspiratory and expiratory. And Betzinger complex, how that fits in there is a little bit more problematic in defining its exact role in generation of rhythm versus pattern. And it certainly seems to be involved in both of those things. Certainly what it does, it has a nice population of inhibitory neurons, inhibitory expiratory neurons that project throughout the respiratory columns and seem to be really fundamental in regulating phase, the proper phase discharge pattern of various groups of neurons. And then pattern. So groups of neurons in the rostral VRG, which is largely composed of neurons that are bulbal spinal premotor neurons, go to the phrenic and intercostal motor pools for generating the inspiratory efforts. And the caudal VRG, similarly organized only for expiratory motor control. So again, expiratory, bulbal spinal, premotor neurons seem to be located there. And then the pontine respiratory group has largely been ascribed in the literature to the caulicular fuse nucleus, which is just at the sort of ventral aspect of the brachium conjunctivum, that's superior to cerebellar peduncle. And the medial parabrachial nucleus is also usually ascribed to having a respiratory function. But when you actually look at where respiratory neurons are located, either defined anatomically by retrograde labeling or electrophysiological recording, you get the most of them are going to be in the caulicular fuse nucleus, and if maybe the most extremely lateral aspect of the medial and the ventral part of the lateral nucleus. These other areas are all very interesting. So ventrolateral pons contains A5, that noradrenergic cell group, and certainly is involved in respiratory control. It hasn't been looked at too much, so we don't have a lot of information on it in terms of its functional role. From a respiratory point of view, it's been looked at much more extensively in terms of a potential role in autonomic regulation and cardiovascular control. And this region here that is interdigitated in the fibers that come from the motor trigeminal and give rise to the trigeminal nerve is referred to the inner trigeminal area. And that's been shown to be involved in the production of a variety of apneas. And it's been speculated, at least by David Carley at the University of Illinois, to be involved in potentially in sleep apnea. That's a bit more speculative though. And then up here, dorsally in the medulla, the nucleus of the solitary tract, which again is the termination site of those afferents, as well as an accumulation of phasically active respiratory neurons in what's referred to the dorsal respiratory group. So without going through all these in detail again, the retrotrapezoid nucleus is that compartment that's just ventral to the facial nucleus, involved probably in chemoreception. There's a little bit of controversy in the literature on just exactly what its role is again. Parafacial respiratory group, certainly being suggested to be involved in expiratory rhythm generation. Betzinger complex certainly controls the phase relationship of various groups of neurons, but is still not well understood in terms of what its role really is in rhythm and pattern generation. Prevesicular complex believed to be involved in rhythm generation, and the rostral and caudal VRGs in terms of that premotor pattern formation. So most of the pattern formation is usually ascribed to occurring in the premotor pools of the rostral and caudal VRG. So coordination between breathing and other functions, a big part of that is likely to occur supramedullary. So the pons certainly is likely to be involved critically in some of it. We've shown, for example, if you block the parabrachial region with mucimol injections, the GABA agonist shuts down the neurons there, that you block the respiratory response to nociceptive stimuli. It's also critically involved in cardiovascular control. And so it's highly likely that that's a primary site of integration between those two. Higher functions like the hypothalamus, and we'll get a little bit into that in a few minutes, hopefully. And that seems to be a region that is much more of an integrative command center for behavioral responses, so is much more likely to be involved in integration of respiration with various behavioral things, such as swallowing, is what I would expect. It's much more at a higher level with lower level, that is brainstem, control of various aspects of it. But certainly from the cognitive aspect, it has to get into higher areas, which then look like it involves hypothalamus in terms of a sort of command structure.
I mean, that's a very powerful separation, and it's probably not higher level. So
No, because animals can do that without being higher level. So you can do a decerebrate animal, and you can feed it that way, in fact. Yes. So how that comes about, it's totally up to grabs in terms of exactly where that would be.
So taking, in terms of swallowing and taking away the cognitive aspect of it, we know we can initiate a swallow by stimulating like the internal branch of the superior laryngeal nerve. So I always wonder, you know, as it's projecting to the nucleus tractus solitarius, what is causing the inhibition? Is it inhibition in the prebutzinger complex that's causing the apnea during swallowing, or what is that relationship? How is that projected?
As far as I know, absolutely nobody has looked at that. So I can't give you a categorical answer. I can tell you, though, that the relationship between afferent input to the NTS and the ventral medulla, so the ventral respiratory column, which is where most of your premotor stuff is going to come from, there are direct projections, both excitatory and inhibitory. And also, in addition, in that processing, it looks like it involves pontine respiratory group area. So that's seems to be necessary for a lot of these inhibitory kinds of effects.
Which part of the VRC column is most densely innervated by cortical inputs?
There isn't a lot of data on that either. So Bruce Nail, many years ago, did some retrograde tracing and stuff. And you know, there's just not a lot of detail on that. We've done a little tiny bit of looking at ventral respiratory column innervation from the cortex. And, you know, it's not very dense. There are neurons there that directly innervate the ventral respiratory column. And that seems to go more heavily, at least in terms of direct projections, to like the rostral VRG level, so the premotor neuron level. But there's undoubtedly, there has to be an issue of control of the rhythm generating circuitry also, obviously. And so much of that, so what's been postulated is much of that gets relayed from the ventral medial medulla. They get the cortical inputs going to the ventral medial medulla that then inhibit more laterally. But again, there's not a lot that's known about that. It's wide open, yeah.
Not to beat the question too much, but I want to flip that question around to you, and I want to ask which areas of the cortex are most likely to project into these. If you retrogradely labeled from ventral column neurons, where do you see
labeling?
Where do you see labeling?
So you see labeling in the medial prefrontal cortex. You see labeling in medial aspects of motor cortex. And you see insular cortex. So anterior bits of the insular cortex. Insular cortex seems to be more related to affective kinds of behavioral related issues as opposed to just general motor commands. But again, that's a wide open area. You know, there's room for some great studies to be done there. All right, what's next? So this is just, we can just go through this quickly. It's the same thing, looking at the ventral respiratory column, retrogradely labeled neurons. This is a parasagittal section, and then it's just showing where the retrogradely labeled neurons are if you do a coronal section through each of these areas. So rostrally through the Betzinger. And you can see the one thing about the Betzinger is it's got relatively fewer neurons, or the much lower density of neurons, maybe it would be a better way to say it, than other areas. Pre-Betzinger is a nice, densely labeled group of cells that's just ventral to that caudal part of the compact or the semi-compact divisions of the nucleus ambiguous, which is right here. And these little white neurons up just dorsal to the nucleus ambiguous then, which would be right here. These neurons here tend to be premotor neurons going to hypoglossal, for example, so cranial-motor neurons. Rostral VRG, all of a sudden now you start to see neurons extending much more dorsomedially towards this solitary tract. It's a much more extensive population. And particularly if you have animals that have intact vagi, you see a lot more respiratory activity there than in the standard reduced preparations that we usually study. And caudal VRG, a similar kind of distribution, just caudal to the caudal tip of the fourth ventricle is usually what distinguishes the rostrocaudal level between the inspiratory and the expiratory populations of neurons there.
Into the ventral respiratory column, there are very, very few neurons in comparison to some of the major nuclei, whether it's locus coeruleus, seventh motor nucleus.
Some of it, there's not a huge number of neurons, that's true. I don't know I can't recall now what estimates there are on that.
From pre-Betzinger, I think we heard about 2,000.
Yeah, I wonder where you heard that from. It's not a lot of neurons. It's not a lot of neurons. And what the real population is that's involved in respiratory control there, I think is still an open question. So I think the numbers of cells you see that you can label with any particular technique is a subset of those that are involved. I can tell you that if you go into, and when you go into a slice preparation and look for rhythmically active cells, and you go into the pre-Betzinger complex, you know, it's not all the cells that are within that anatomically described or circumscribed region that are respiratory. But if you go to an in vivo behaving animal, well, forget the behaving, anesthetized animal, and record in that same area, every neuron you record is respiratory over an extent that is at least 500 microns, dorsal to ventral. Even in a rat, it's got to be 500 microns. You eliminate a lot.
Not just anatomically. That sounded like what you just said, functionally.
Functionally, you've lost a lot of neurons. And the other thing that can be a little misleading is when you look anatomically, histologically at the ventral respiratory column, what you find, and particularly in the smaller mammals, that neurons are clustered. So there's large bundles. What you don't get from these histological slides is there are a lot of myelinated fibers running through here, and they run through in bundles. And sort of in between the bundles, you have a cluster of 3 or 4 cells, all of which are presumably respiratory. And it can make it really hard when you're trying to isolate the recording from a single neuron, because you got 3 or 4 cells that are cheek by jowl and in a small mammal. And so you got to use a pretty high resistance electrode to get a good isolation of the single unit in the smaller. Yeah, no, that's right. It definitely reduces the productivity.
Frustrating with regard to sheets of blood vessels.
And that's true too. And you can see that. So what Don's referring to is if you look at anatomically here, we're on picture. Oh, no, you can see it actually a little bit right here. So all these little green spots are clusters of cells. And you can see it's not the blood vessels you're seeing here that are red. So red in this case is the NK1 receptor. But you can see how you get lines of cells. And the blood vessels come up from the ventral surface and run up towards in a sort of dorsal-medial direction. They come from ventrolateral and run up dorsal-medially. And the cells all sort of line up along that sort of trajectory. You see the same kind of thing. And so I just brought this slide in particular because you always hear about NK1 receptors and how they mark the pre-Betzinger complex, and they do, sort of. And so it's important to sort of get a real, I think, flavor of what that means. So if we look at this slide, the top part is a parasagittal section, and you can see the compact division of the nucleus ambiguous, tons of NK1 receptors. It's certainly the densest NK1 receptor population in the neighborhood, by far. And the facial nucleus, pretty much devoid of it. The lateral reticular nucleus, which is a pre-cerebellar nucleus, is also pretty much devoid. But here, this rostral part of the rostral VRG actually has lots of NK1 receptors. And it's, to refer to the pre-Betzinger as having NK1 receptors is absolutely correct. It's enriched relative to things around it. But you can see there's not a really bright NK1 receptor line here, and followed by absence of NK1 receptors as you move rostral and caudal. It's a much more subtle effect. Parvalbumin here, the green, is actually used to mark bulbar spinal neurons. So 2/3 of the respiratory premotor neurons contain parvalbumin. So that's how we were able to identify the rostral VRG and the caudal VRG. But also to keep in mind, when people talk about NK1 receptors and marking the pre-Betzinger complex, it's important to realize that when it was originally used in the first paper from Jack Feldman's lab by Paul Gray and company, it was that NK1 receptors in combination with mu opioid receptors be marked. Yeah, and so that's a part that's always left out. And so there's not a really great marker yet for pre-Betzinger neurons. There's several markers that you can hear about that are out there. None of them are really super terrific.
Pre-Betzinger is, how many different cell types, expressions would you say are up there? I mean,
I missed the first part of your question.
So I've heard a lot of reports from, X number of inhibitory, X number of excitatory, diverse populations in the pre-Betzinger, this function, that function, and overlaying just mishmash of cells. How many different populations would you say are there? How many different phenotypes do you think you're going to find in that pre-Betzinger area that everybody looks at?
So I think the answer to that question is in the sort of eye of the beholder a little bit, and what you're going to use to define the different populations. So if you go on a strict classification system based on discharge patterns in an in vivo animal as opposed to an in vitro slice, in an in vivo animal, typically you come out with approximately 6 different subgroups of respiratory neurons. And add on to that, and that doesn't really include much in the line of some of the phase spanning neurons. So you could probably increase that by another 2 or 3 sort of globally. And concentrated within the pre-Betzinger complex, you'd probably find 4 or 5 of those based electrophysiologically. But now you start throwing in functional roles and what the transmitter is they contain, et cetera. And there is no estimate on that. I mean, people just don't even try, really, on just how many groups there are in that region. But certainly, and it's, one of the ways it was defined initially, one of the primary mechanisms of defining it was it lacked bubble spinal premotor neurons. That's a slight overstatement, but it's largely true. But it's not so clear that it doesn't contain premotor neurons going to the cranial nerves. And so just dorsal to that population, for sure, there's a good population of hypoglossal premotor neurons. But there's probably also premotor neurons for many of the cranial neurons that are located within that region also. So I can't answer I'm coming up with a lot of non-answers, but that's good. Well, maybe it's not good, but it's the way it is. All right. So speaking of premotor neurons, here we have a depiction of the elements that we know about that are certainly involved, and we ascribe to pattern formation or pattern generation as opposed to rhythm generation. And so premotor neurons, expiratory ones, and so you can see this funny looking triangular thing is supposed to be a premotor neuron. It has an axon that's excitatory, goes down to the thoracic spinal cord and innervates expiratory intercostal motor neurons. And it does not seem to give rise to axon collaterals, interestingly enough. Unlike, if you go just rostral, you have bubble spinal excitatory neurons that give rise to axon collaterals, and so you get this integration going on within the rostral VRG, but some of these collaterals go beyond the rostral VRG and project even more rostrally up to the pre-Betzinger complex, for example, as well as the cranial motor neurons. So there's a fairly extensive arborization of some of these inspiratory bubble spinal premotor neurons.
Is there a consensus on how many of those project ipsilaterally versus crossing in the bella and project on the other side for the premotor?
A number I can't give you, but and it depends on the species. So in the cat, the vast majority are contralaterally projecting. In the rat, I think a very high proportion project bilaterally, and there can even be a little bit of a dominance ipsilaterally. But you get a bilateral projection in the rat for sure. Mouse, nobody's looked at at all. Yes.
Doctor, I can see from your sketch that practically all the respiratory column somehow is projecting directly to the spinal cord. And,
no, actually, let me just, before you go finish, I'll let you finish your question, but the round circles here aren't supposed to be neurons. Those are terminals. Those are inhibitory terminals. So we don't have anything coming from, the red here is the pre-Betzinger complex. We're not showing anything projecting from the pre-Betzinger complex to the spinal cord.
those projections, they are mainly inhibitory, I suppose, right? Other than the rostral ventral respiratory group.
Many of them are inhibitory, yes. So anything arising from the Betzinger complex, there's a large component of inhibitory bubble spinal neurons arising from the Betzinger complex. For expiratory neurons, there's also bubble spinal expiratory neurons that really extensively arborize throughout the VRC. And those are located much more extensively. So it's shown here, this one is shown on the border between the rostral VRG and the caudal VRG. They extend fairly rostral, well, pretty much anywhere you want to look in the VRC, you find some of those neurons. And the difficulty, though, with making too categorical statement about them is that many of them are actually cranial motor neurons. And so it's an extraordinarily rare study. In fact, I don't know of any real study that really can say, when they record from an expiratory decrementing neuron, which is the kind of pattern these have, or a post-I kind of activity, whether they are categorically interneurons, motor neurons, or bubble spinal premotor neurons. So you get into that problem. But certainly, those neurons are inhibitory and project through both the brainstem and the spinal cord. All right, NTS. So the dorsal respiratory group, it's kind of fallen by the wayside and hasn't got much attention paid to it lately for a number of reasons. One, you don't really find one in the in vitro prep, and most labs these days are doing in vitro. And in addition, there's a distinct species difference. So it used to be the NTS was a big deal when people studied cats because there were lots of respiratory neurons, basically active respiratory neurons in the NTS of the cat, even in vagotomized animals, so they weren't just getting afferent input that was phasic. In rats, that's much harder to define, and it's pretty clear that there is a dorsal respiratory group, but its exact location and distribution of its neurons, there's only a couple of papers that have really report on it in any kind of detail at all. So it's not very well understood. Nevertheless, there are some bubble spinal neurons. So the dorsal respiratory group in the cat is mostly bubble spinal premotor neurons. In the rat, it's not really clear what they do, other than they do project to the spinal cord, at least a subset of them. In addition, obviously, just given where these neurons are located, you'd expect that many NTS neurons project to other respiratory groups and provide that sensory input that's coming from the vagus or the glossopharyngeal nerves or the trigeminal nerve or even the facial for that matter. And one other point is chemoreception. So it's clear that neurons in the NTS have intrinsic chemosensitivity. That's pretty clear. How important that is for respiratory control is much less clear. But I'm sure you got a much more detailed discussion of that from Gene Natty when he was here. So I'm going to just leave that for the moment and move on. And what I'd like to do is take an example of a particular reflex. And that's the reflex that's evoked by activating slowly adapting pulmonary stretch receptors. So if you activate slowly adapting pulmonary stretch receptors, you get a couple of effects. You terminate an ongoing inspiration, and if you maintain that activation or lung inflation into expiration, you lengthen the period of expiration. So you get fundamental changes in respiratory rhythm. And so what are the pathways that subserve that response? So slowly adapting stretch receptors, this is supposed to represent the vagus nerve with the jugular ganglion and the nodose ganglion. It has two ganglia. And slowly adapting stretch receptors are located in airway smooth muscle and then project up, terminating in the NTS. There's another group of receptors which referred to as rapidly adapting receptors, which tend to have, this is an oversimplification maybe, but their reflex effect tends to be much more one of inspiratory facilitation. So it can give rise to size augmented breaths, for example. Also located in the airways, but their afferent terminals are located in airway epithelium, as well as, in particular, down in the vicinity of venules. So airway congestion, inflammation of the airways can give rise to activation of rapidly adapting receptors. Maybe they're more normal stimulus as opposed to lung inflation. Their differentiation can go back to the sort of original description of them from Knowlton and Larrabee in 1946. Slowly adapting receptors, all we're showing here is the change in lung inflation. And as you inflate the lung, you activate slowly adapting receptors, and as you maintain lung inflation, they tend to have a maintained discharge pattern, whereas rapidly adapting receptors, you do the same reflex response. They adapt rapidly and terminate their ongoing activity, despite the fact that the inflation is being maintained. So electrophysiologically, they're different. And activating of the slowly adapting stretch receptors produces the Breuer-Herring or the Herring-Breuer reflex and terminates an ongoing inspiration. And as you see here, so what's on the bottom here is just lung inflation, so the pressure used to inflate the lungs. And if you maintain the lungs inflated, you shut off phrenic nerve activity, by and large, or at least produce a much lengthened expiratory duration. And so if we want to understand sort of the coordination of respiratory control, one thing to do is sort of work out the circuitry that gives rise to this. And how do you do that? Well, first off, actually, I forgot I was going to show you this, is a slightly more subtle nature of the pulmonary stretch receptor reflex response. And so what's done now, this is actually a dog in this case, and using a ventilator to slowly deliver a lung inflation during expiration. And what you find is, if you look at phrenic nerve activity, it's shut off during this inflation, during the expiratory activity. But if you record those in the rostral ventral respiratory group, those inspiratory bulbospinal premotor neurons, what you find, excuse me, this is the caudal ventral respiratory group, the expiratory neurons, you find that at low pressures, you actually have an increase in their discharge, which you might expect that would facilitate expiration. But as you increase their activity, you actually inhibit their ongoing activity. So from a reflex point of view, or a teleologic point of view, which is always dangerous, why would you want excitation at low inflation pressures? And if you think about it, what is this reflex doing? It's telling the brain the lungs are inflated. What you got to do is get the air out. So thinking simplistically, what you want to do is activate expiratory muscles, drive that air out. And if you have low inflation pressures, it's saying that's exactly what you do. But at higher inflations, you get a lengthening of that expiratory period. That's true. But you're actually inhibiting those premotor neurons that you'd expect to be activating the expiratory muscles. So teleologically, I don't have an answer for you, other than to say, what you could imagine is that at high lung volumes, you've got a lot of recoil force, and you've got a lot of force that's innately driving the air out. And if you're familiar with doing maximum flow volume curves, you know that after some point, increasing the force of compression of the lungs doesn't help you get air out any faster. So you might as well conserve the energy and allow the elastic recoil to drive the air out. That doesn't work once you get to the smaller lung volume, and now you got to recruit expiratory muscle to get the air out. So that's a teleological argument for whatever it's worth, but it's the only way we can sort of think about, you know, how this thing makes sense. But it is the way it is. So mapping out the central pathway for this has been kind of fun. And so this is just a horizontal section now through the brainstem, and the vagal afferents have been labeled with a fluorescent tracer. Doesn't particularly matter what it is, it was di-eye. And you see now you get fascicles of the vagus nerve entering the solitary tract, and they course caudally, and they arborize all over the place in the NTS, and even project to the contralateral side. An important point for understanding sort of lateralization of these reflexes. You do have afferent input, particularly from the vagus, that goes to both. And when you go in there, you can record either from the primary afferent axon and fill its axon, so you can see the terminal arborization, and that's all that's been done here. So here is filling of a primary afferent axon. You record from it, and you can identify it. This is actually a cell, but the afferent axon does the same kind of thing. And you look at airway pressure, and as you inflate the lungs, you activate these afferents, so you get a discharge pattern. To maintain that inflation, you have a maintained discharge pattern. And so you can fill either the primary afferent, and you can get its termination, which ends up being sort of in the interstitial nucleus, intermediate, and ventrolateral portions of the NTS. So, in other words, in the region of the tract extending fairly much to the cell population that's actually located right within the tract itself and extending ventrolaterally from that. And what's traditionally thought of as the ventral or the dorsal respiratory route. And if you record the cells in that region, they're referred to as pump cells, named by Albert Berger, because they inflate or they fire every time you inflate the lungs with the pump. And you can fill those and look at their axonal arborizations. They arborize, they send an axon down to the ventral respiratory column, and they extensively innervate then rostrocaudally in the ventral respiratory column. And you can follow their axons for a fair distance in the ventral respiratory column. And what you can't do is follow them all the way up to the pons, but they go all the way to the pons. And this is just showing a slide from Azura and colleagues who then antigeromically mapped the trajectory of these pump cells projecting all the way up to the rostral pons. And so if you think about it, what would the pons be doing getting these pump cell inputs? What you want to do is shut off inspiration. Well, coordination with other things could be an important role. Also, if you think about it, lung inflation actually reduces sensation of dyspnea. And so you've got to get somehow that afferent signal that's related to lung inflation going more rostrally. A stepping point for more rostral projections is, in fact, the rostral dorsolateral pons and the pontine respiratory group. So that's probably why they go up there. And many of them are, in fact, inhibitory. So this is another slide from Azura, just showing a recording of a pump cell and then filling it with dye and doing immunocyto, or not immuno, what am I trying to say? In situ hybridization, and looking for GABA and glycine. Actually, this was immuno because he did GAD67. And what you do find is that many of them are inhibitory. And one of the things they do is if you fill them and look at them and see where they go, they actually inhibit the cells that receive the input from rapidly adapting receptors. So activation of slowly adapting receptors, which facilitates expiration, inhibits the cells that are going to facilitate inspiration. Physiologically, again, kind of makes sense. They also project, as I just mentioned, heavily to the ventral respiratory column. But if you recall, one other point is just to show if you actually record from the ventral respiratory column, activate vagal afferents. And one of the sort of fortuitous aspects of this is slowly adapting pulmonary stretch receptors tend to be the largest afferent in the vagus nerve, so they can be activated with the lowest electrical threshold. And so you can activate them not completely selectively. RARs, or rapidly adapting receptors, overlap with them. But you can get a fair idea of their synaptic input by using very low intensity currents and looking at the response pattern of neurons. And in this case, looking at the VRC, you find that most of the input from these cells is inhibitory. However, there is a subset that is excitatory. And again, these are those EDEC cells. So the subpopulation of those cells that fire during expiration with a decrementing pattern, many of which are those bubble spinal premotor neurons that we talked about. And they receive an IPSP. Many of these cells are inhibitory and project throughout the VRC and can provide that inhibition of expiration. So, in summary for that, we have the pulmonary stretch receptors, just slowly, SAR is slowly adapting pulmonary stretch receptors coming in, terminating in the NTS, and then they're going to activate either inhibitory pump cells, and the interesting aspect here is that the inhibitory pump cells project throughout the entire ventral respiratory column and on up to the pontine respiratory group. So they inhibit cells, they provide an inhibitory input to each subcompartment of the ventral respiratory column. The pre-Betzinger complex, the Betzinger, rostral, and caudal VRG all receive strong inhibitory input. If you look at excitatory projections, they're much more selective. They actually tend not to hit the rhythm generating areas. So they still project to the pons, or appear to project to the pons, but they target in particular the rostral and caudal VRG and the Betzinger complex, skipping the pre-Betzinger complex. And in fact, it's probably not even the Betzinger complex they hit, but rather the retrotrapezoid nucleus. All right, so we're at about 5 minutes to 5. So I can stop right there, which would be fine. That's a nice convenient stopping point, or we can do some optogenetics, or we can talk about carotid chemoreceptor pathways. So I'm open to whatever people would like me to do.
Pick one.
Pressure on me? No, I'll go optogenetics any day of the week. What are you going?
Can I ask a question first?
Yeah.
So the NTS projections you're showing there, those are all based on the intracellular fills and mapping. We heard a really interesting presentation recently about vagal stimulation as a rehabilitation tool. So the NTS cells, they've got to be projecting like throughout that bay nuclei and up to the brain. And what do you know about the other places that NTS is projecting?
Other places that NGS, I can just, what I could do maybe, I don't have them all down here. But what you can do, actually, I guess I don't have the full thing in here, do I? But so this is for the optogenetic bit and leptin. So leptin gets involved in a lot more hypothalamus, et cetera, areas. And talking about mechanisms for relating respiration to metabolism. And it looks like, so for example, the pathways that are activated by exercise, which are not understood at all, either from a physical point of view or from a respiratory control point of view. But they seem to activate many of the same pathways that are activated in stress responses. So one of the ones that's classically used is an air puff stress. And it gives rise to activation of hypothalamic regions, in particular the dorsal medial hypothalamus, and the lateral hypothalamus, the posterior hypothalamus, and pericortical region. Periaqueductal gray is another area that gets fairly strongly activated, and the parabrachial region. And so parabrachial, from a leptin point of view, it's particularly the superior lateral nucleus. But undoubtedly, it will expand out for other modalities. So I lost track of what the exact question was now.
Just places that vagal afferents would ultimately project to that could facilitate motor rehabilitation.
So I would guess that it's activating some of those pathways. Other ones that I'm not mentioning or I didn't mention also go on up to the insular cortex, which brings into play the amygdala, and that's much more of the emotional motor system, as it were. So I'm not sure how much that would play into it, but certainly areas up in the hypothalamus, in particular, I would think would be particularly involved in that. Yes.
You sort of answered that question. I know from the third order projections that goes to areas in the coral ponds called the parasitic cellulars and the nucleus plasma hypervelocity. And from there, the direct projections to the lapping and the locus coeruleus. And people hypothesize vagus nerve stimulation is causing excitation of these neurons through caudal ponds, through hypervelocity and parasitic cellulars, which then go on to promote this motor recovery change.
Well, it's now 5 o'clock, so I know it's scheduled to end, so. Oh, is that right?
There's beer waiting, so somebody's in. You want a beer or you want to talk?
So I'm easy. I mean, I'm totally happy to do whatever people would like me to do here. On chemoreception, or I can skip that.
Chemoreception would be the most good for people here.
Oh, yeah? Okay. So obviously people here know a lot about chemoreception and the respiratory response to it, as well as, in particular, this is just showing the response to stimulation of the carotid sinus nerve, which carries carotid chemoreceptor afferents and the phrenic response to it. And doing the same thing that we did with the vagal afferents, but now starting with the carotid body. And so putting di crystals in the carotid body and waiting for a couple of weeks for it to transport. Centrally, you see a much more restricted projection pattern, but it still goes bilaterally. And if you look at it in detail, you get the axons coming in, and they don't really arborize until you get down well into the region that's rostrocaudally covered by the area postrema and down into the commissure NTS, just caudal to the area postrema. And what I'm going to tell you is that it gives rise to projections that activate second-order neurons that project, as I alluded to for the excitatory projections from the pump cells, all the way through the VRC on up to the rostral dorsolateral pons. But now we really stress that those projections appear to skip the pre-Betzinger complex and the Betzinger complex and do go to the parafacial region, which would be more likely to be the retrotrapezoid nucleus in this case, and the rostral VRG and caudal VRG receive strong excitatory projections. And so just looking at that, this is just showing we can actually record from cells in the NTS that receive that primary afferent input. And so in this case, the animal is given, it's just switched to 100% nitrogen in the inspired port of the ventilator. We're recording the neuron, and its firing rate goes up, and it fires. Its maximum rate is pretty much what you expect for neurons in the carotid body, actually, or cells in the carotid body that give rise to the primary afferent, with peak firing rates that are around 15 hertz or a little less. And you can fill these just juxtacellularly is the easiest way because they're small cells, and do a reconstruction of these cells. And you can also do that in combination now with labeling the primary afferent terminals, as well as retrogradely labeling from the VRC and the juxtacellular labeling. So triple labeling, looking at the integration of primary afferent input to cells in the NTS and their output. And so in this case, this is a coronal section through the nucleus of the solitary tract. Where's my mouse gone? And you've got arborization then of the red terminals. So these are the primary afferent terminals. Juxtacellular labeled cell is shown here in green. And you can see a blow up here with a single slice with a confocal scope showing terminals that appear to be on the dendrites of this cell. Quantitatively is that there are very few carotid body afferent terminals on any given cell. So we never saw more than 2 or 3 terminals on any given cell, or rarely saw more than 2 or 3 terminals. Given the limitation on that, though, that we can only map with the filling, we can only map maybe the first 100 microns or 150 microns of the cell dendrites. So on longer dendrites, it could receive more terminations of the primary afferents. But anything on the cell body or with 100 to 150 microns of the cell body, we should be able to see. And then this is a fluoro gold labeled cell, which is the same one that we see here. And so it shows that this cell does in fact project to the ventral respiratory column. So it's simply a retrograde filling, showing that these cells do in fact project to the ventral respiratory column. And this is just a bit more of the same kind of thing, only looking in a bit more detail. So looking at single confocal slices and looking at terminations on NTS cells. You can see blue labeled processes with red terminals on them. And again, now what we've done is try to look at differential projections. So we use 2 different tracers in different parts of the ventral respiratory column and retrograde labeled cells. And we find that we get divergence of single chemoreceptor axons then to multiple levels of the VRC. And this picture here is just showing now the coronal section to the caudal part of the NTS. So the area that we're talking about of particular interest is this area that's just ventral to the gracile nuclei here and dorsal to the dorsal motor nucleus of the vagus and the hypoglossal nuclei. At what?
The carotid chemoreceptors do not project to the pre-vascular complex. If I take an in situ rodent that I work with, and I give a little bit of really dilute cyanide, and I stimulate these, the first thing that happens is my rhythm goes nuts. It fires faster and faster. Then it tapers off, and it's a really acute hypoxic response. It increases the speed of rhythmicity. How am I accomplishing that if my chemoreceptors are not exciting something in my...
At least direct. So my argument would be they don't go there directly. You do have possibles for indirect projections. Where is that picture of the bubble spinal neuron, wherever it was? So you do have the indirect projection. So you have these cells that arborize rather extensively that also do go to the pre-Betzinger complex. And the retrotrapezoid nucleus, which definitely receives these, projects heavily to these aspects of the VRC. And so one has to ask a little bit about what the real role of the retrotrapezoid nucleus is, because the retrotrapezoid nucleus is kind of interesting in as much as it doesn't really have very much in the line of spontaneous respiratory activity. It has some. It's got an EDEC kind of activity in particular. So it seems to receive an excitatory input from EDEC cells. But on top of that, it receives slowly adapting pulmonary stretch receptor. It receives C fiber from the vagus. It receives rapidly adapting receptor input from the vagus. And it seems to be a central chemoreceptor, at least by the current thinking. And so it seems to be integrating a lot of these different things. And so what is its real functional role, and how is it tying all these things together? And part of the problem is that in vivo, it's darn hard to look at. The cells are small. They're, only 100 microns or so from the ventral surface of the brainstem. So if you're coming in dorsally with an electrode, for example, it is really hard to hit those suckers, and they're not densely packed. So on any given track, you might not hit any. So it's an extraordinarily low yield set of experiments. So my hat's off to Patrice for doing most of the work on the in vivo RTM, because those are hard experiments to do. All right. But that pretty much says what I wanted to say about the chemoreceptor, peripheral chemoreceptor. So in terms of the leptin stuff, it's a much more conceptually oriented aspect of this. But this goes back to when I first met Gordon, and this is actually one of the slides he drew for me. Way back in whenever, 100 years ago. And matching of metabolic rates and ventilation. So you get this parallel increase that you guys know only too well, giving rise to not necessarily an exact isocapnia, but you get a matching of ventilation to CO2 production that produces a progressive change in ventilation in proportion to the change in metabolism. And the question is, how the heck is that done? Nobody really knows. But there are a few observations that could be particularly interesting. And one of these is just, if you start looking at exercise, and there's nothing in the literature at all, but psychological stress, as I mentioned just a few minutes ago, seems to activate many of the same neuron populations that appear to be activated in exercise. The exercise data, there isn't a lot of good exercise data. Obviously, recording in an exercising animal is a challenge. So there's not a lot of data there, but there are some CFOS studies, et cetera, looking at activity changes, and so there's a lot of overlap in the pathways that get activated. And of these places that get activated then, ones that come up over and over again are the periaqueductal gray and the dorsomedial hypothalamus in particular. And those have been mapped out as responding in particular to a wide variety of different psychological stressors, which are then rooted by a number of different pathways that are sort of outlined here, down to the periaqueductal gray. And then the output of the periaqueductal gray goes to the dorsomotor hypothalamus, a dorsomedial hypothalamus, as well as down to the medulla itself to activate respiratory and autonomic neurons. And the CNF here is the cuneiform nucleus, or peduncular pontine tegmental nucleus. So the groups that get involved then are, in particular, from an autonomic point of view, they talk about the rostroventral medulla, caudal ventrolateral medulla, the NTS, parabrachial KF. We've certainly heard about that in respiratory control. The one I just mentioned, periaqueductal gray. A couple of hypothalamic regions, the paraventricular nucleus, which is known to strongly influence autonomic function, but also activating neurons there has strong respiratory effects. Dorsomedial hypothalamus and the parafornical region of the hypothalamus, as well as included here is the amygdala, that sort of output nucleus for the emotional motor nerve system or the limbic system. And so if we look at coordination of these things, and if you just disinhibit activity in the dorsomedial hypothalamus, you see tremendous coordinated changes in cardiovascular and respiratory output. So this is just injecting in the dorsomedial hypothalamus bicuculline, the GABA antagonist. So you get an increase in activity of any neurons that are chronically inhibited, which appear to be many of the neurons in the region of the dorsomedial hypothalamus, giving rise to changes in arterial pressure, heart rate, renal sympathetic nerve activity, to more generally look at that. But in this case, most of the respiratory change is a frequency change as opposed to an amplitude change, which is kind of interesting. And this is just showing those effects now at a slightly higher time sweep speed. Same injection, just looking at a higher sweep speed, so now you see individual clinic nerve bursts, for example. You can see how much that increased the burst rate. And if you look at arterial pressure, you can see how much that has changed also, as well as heart rate. So this diagram again. It's saying that between the periaqueductal gray and the dorsal medial hypothalamus, you've got some sort of a coordinating system for producing this integrated response. How is it done? And could this have anything to do with other forms of respiratory stimulation, such as that comes by with leptin? I think I deleted the slide on this, did I? Yeah. So the story of why to look at leptin at all sort of comes from the stuff from Chris O'Donnell at Johns Hopkins and their group that did a bunch of studies way back late 1900s, early 2000s, and looking at a mouse model of obesity. So the OB mouse lacks leptin. It was the first genetically modified mouse ever created in the lab, I think. And it's been pretty well characterized, but OB mice lack leptin, and they become obese. So leptin is sort of thought of as produced by adipocytes and a satiety hormone. So you consume foods, consume calories, you produce more leptin, release more leptin, and it produces satiety. So it was tried at one point as a diet-inducing drug to lose weight. However, large levels or high levels of leptin actually produce leptin resistance, just like you get insulin resistance. And so it didn't work in humans, at least, as a mechanism for weight loss. Nonetheless, you get these obese mice, and they hypoventilate rather substantially. So they have a 10 torr or greater increase in arterial PCO2. So pretty darn substantial. So what O'Donnell and company did then was to put Alzette mini pumps or the like into these animals and administer leptin at low levels over 3 days and then measure them again. And what they found was, yes, there was a little bit of weight loss, but in 3 days you don't lose a whole lot of weight. But breathing came back to normal. And as much as arterial PCO2 was now normal, or within the normal range. So there was a substantial increase in ventilation, matching their metabolic demand. And it seems to be provided by leptin somehow. And so where are the regions that leptin acts? So leptin has several target regions. Two of them you'll recognize from respiratory anatomy, the nucleus of the solitary tract and the parabrachial nucleus. Again, specifically the superior lateral subregion of the lateral parabrachial region. But in addition, it's got a bunch of areas in the hypothalamus, most notably the arcuate nucleus and the dorsal medial hypothalamic nucleus. I can almost not say anything that starts with DM other than dorsal motor, thinking of dorsal motor nucleus of the vagus. I'm so fixated on that. But anyway, I suppose, but I never think of that. So those in particular seem to be of interest. And so our goal was to sort of look and see at how these signals all match up and whether then these nuclei that are also activated by stress responses and potentially during exercise might be some source of neural signals that would help relate breathing to metabolism or the metabolic demands of the animal. And just giving leptin IV, so this is an acute in an anesthetized animal, and giving lactated ringers containing leptin, and looking at integrated phrenic, you see over a period of time it goes up. So it's an infusion for an hour, and you get a long-lasting increase in phrenic amplitude. And that's true in a large number of animals, and it seems to be that it peaks out at about a 50% increase in minute ventilation in these animals, which is fairly substantial. You also have, and it's been documented by other people, you get an increase in the response to CO2. So CO2 chemosensitivity, the slope gets greater. And just recently, David Fuller with the Yowan Company published a paper looking at where the leptin may be working. And they did it in an interesting way. They did the leptin injections into the fourth ventricle or the lateral ventricles. And what they found was if they injected leptin into the lateral ventricles, they got an increase in both breathing magnitude, and so phrenic output, and airway tone. So airway tone increased, reducing the incidence of obstructed sleep apnea. On the other hand, if they injected leptin into the fourth ventricle, they got an increase in breathing and not in airway smooth muscle. It took me a minute there. They didn't get an increase in the tone in the airway muscle. So they were looking at NTS as probably the site where they get most of their response on phrenic motor output. And fourth ventricle sites, so potentially the dorsal medial hypothalamus, is a site where you may get that change in airway smooth muscle tone. Well, we did something kind of similar a little bit. We did anemically decerebrated animals, rats, so I won't spend a lot of time on this, but what we did was we injected polyvinyl siloxane bilaterally into the carotid arteries. Polyvinyl siloxane is the dental impression material you get. And so you use a low viscosity polyvinyl siloxane. It will fill the carotid arteries, obviously, and the circle of Willis, and the largest blood vessels coming off the circle of Willis. But if you carefully control the amount you inject, you don't fill up the more caudal aspects. So the caudal aspects of the posterior communicating artery aren't filled in the basilar artery. So what are we looking at here? The top picture is just obviously the entire ventral surface after one of these experiments. The purpley pink bits are polyvinyl siloxane. ICA is the internal carotid artery, medial cerebral artery, posterior cerebral artery are all filled with polyvinyl siloxane. The basilar artery now, so these animals post the experiment are perfused with a solution that contains Evans blue and what was the other one? There was two, Evans blue and it might have been ponamine, I'm not sure on that one. Anyway, two blue dyes. And so wherever the brain looks blue, black, and white means the blood vessels have been cleared of red blood cells, so the perfusion got there, and left behind some of the dye. The pink means, one, we've got polyvinyl siloxane, and two, more importantly for the pink part of the cortex here, for example, it's because when you do the post-experiment perfusion of the animal, the perfusion doesn't get to the cerebral cortex, for example, and so it doesn't wash out the red blood cells. So the pink is the red blood cells that are left behind. So it's saying that the forebrain, including the hypothalamus, which is right here, is nice and pink, and you don't get a perfusion of the hypothalamus. So the hypothalamus is dead, and we do EEGs to, in fact, ensure that the forebrain is non-functioning. And you can look at a little bit more detail at just where the transition is from perfused to non-perfused, but clearly it's caudal to the hypothalamus, seems to go through the sort of mid part of the inferior colliculus. But all of the parabrachial region and medulla seem to be well perfused. And if you do the repeat experiment where you give systemic leptin, you still get about the same increase in minute ventilation that you got in the or the other group of animals that had an intact forebrain. So it suggests that the hypothalamus at least isn't required for this response. So obviously NTS is a target, so we did a little optogenetics. So we use a leptin-free mouse that expresses channel rhodopsin now, flux channel rhodopsin, in the leptin receptor neurons. And one of those populations is in the NTS, and so this is just immunostaining for, in fact, GFP. So the animals that have channel rhodopsin in their neurons also express GFP. So we have a GFP promoter there also. And so we can immunostain for that, and you can see where it is. And this is, in fact, just a blow-up of those cells in the NTS that are positive for GFP, or by extension, the leptin receptor and channel rhodopsin. And so if we optogenetically activate those things, we in vitro see nice responses. So this is in a slice. And we just see that depending on how much light we give, we get a bigger and bigger response. And that if we put leptin on itself, we get a delayed. It takes time for leptin to exert its depolarizing influence, but it will depolarize these neurons over a period of time. And now in vivo, if you do it, and we can put the laser or the fiber containing the laser light just on the dorsal surface of the medulla. So you don't even have to stick it in the medulla. Activate the leptin channel rhodopsin neurons, and you get a pretty substantial increase in breathing. And almost all of it is related to an increase in tidal volume, with relatively little change in respiratory rate, and for that matter, little change in heart rate. Even though leptin receptors in the NTS have been linked to increases in arterial pressure, sympathetic outflow, and increases in heart rate. But we didn't see it. But everything isn't quite as simple, and this is just to show that there are complex issues going on here. And so in many cases, what we see is, as shown here, we give multiple short pulses in this case, we're using an LED to activate the channel rhodopsin. And we give relatively short pulses of it. During that pulse, we actually get a decrease in respiratory frequency. And so this first one is just blown up here to show the decrease in frequency with no real effect on amplitude during that short burst. But over a period of time, as we do multiple, we get a progressive increase in the amplitude that outlasts, obviously, the stimulus period by quite a bit, and then gradually recovers back down to control. So how do we explain this? First off, clearly we like the idea that activation of channel rhodopsin in these neurons produces the increase in breathing, but there are multiple different subsets of neurons in the NTS that have leptin receptors. And so several of them are likely excitatory and contribute to respiratory stimulation, but some of them could in fact be inhibitory. And so there's three populations that have been well characterized. Now, one contains a glucagon-like peptide, or the proglucagon, and it's the only place virtually the only place in the CNS that can change this particular peptide. And it's in a subset of leptin receptor neurons in the NTS. Another group contains CCK, and a third group contains POMC, or pro-opiomelanocortin, so enkephalin precursor. And GLP-1 has been shown to be involved in cardiovascular control. So we thought we'd look at it, see what we could get in terms of mimicking the response to leptin receptor neurons. So Ida Llewellyn-Smith from Australia has shown that many of these GLP-1 neurons, or glucagon-like peptide neurons, project to the ventrolateral medulla and do produce changes in cardiovascular control. So we simply injected an agonist, which is a synthetic GLP-1, into the ventral respiratory column, and when we do that, we get a nice little stimulation of breathing. Certainly amplitude increases without that decrease in respiratory rate. So we think at least the stimulation of the amplitude is probably mediated via these neurons from the NTS. But there are several other populations, as we already mentioned, and what happens with them. So the dorsal medial hypothalamus, if we activate those neurons, we still get an increase in respiratory motor output. Again, almost exclusively by an increase in the amplitude of the phrenic nerve bursts. But in this case, actually, as you see here in this example, we actually do have something of an increase in respiratory rate also. Injection of, in this case, instead of the optogenetics, we actually injected leptin into the parabrachial, superior lateral parabrachial nucleus. It also produces an increase in respiratory motor output. And go one step further, do it in the rostral VRG. And there are some leptin receptors that extend all the way down through to the ventrolateral medulla. And so it wasn't really thought they would probably do much to breathing, but when we put our fiber to activate the channel rhodopsin in the VRG, we do find we get an activation of rostral VRG neurons and a modest increase in respiratory output, and that can be matched by an injection of leptin directly into the rostral VRG. But just to show that not everywhere that you go then would produce that stimulation. In this case, it's activating cholinergic axons that are in the lateral hypothalamus. So these neurons probably inhibit orexin-containing neurons in the lateral hypothalamus. And when you turn on the channel rhodopsin, then you're going to turn off the orexin neurons, and we get an immediate decrease in phrenic output. So the bottom line is we think there's sort of a hierarchical control system. And so activating leptin receptors in any of these nuclei that are known to be involved in respiratory control at some level, and certainly involved in other aspects of metabolic regulation, and are also cells that are activated by various stressful situations, and we hope ultimately to test whether they're activated in exercise, but seem to be involved in this regulation of breathing that could contribute to producing a proportional increase in metabolism and breathing. So I think I'll just leave it there. We won't bother walking through this. Yes?
The leptin receptors are also located on respiratory motor neurons.
No evidence whatsoever. There's no evidence that they're in, for example, we haven't looked in the phrenic, for example. So I can't say categorically. I think there is evidence that there are some in the spinal cord. I don't know of any evidence saying they're on motor neurons, and certainly when we look in the brain stem, we don't see them on hypoglossal neurons, which would be the most obvious, or facial motor neurons.
So maybe some other spinal interneurons.
They could be, yes. They could indeed. That could be yet another mechanism for it. And in this hierarchical control system, we think as you go up the system, we're getting more and more integrated in the responses and the overall control. But as with many of these feedback systems, you have several levels of control. And feedback from chemoreceptors doesn't just go to premotor neurons or motor neurons. It goes all the way up the system. Yes.
Read an article late last year that was looking into the communication between appetite and satiety and the microbiome in the gut. It was suggesting that there were signals being sent by different populations of bacteria that once they reached a certain number of replicants, say 1 billion replicants, they would send signals to release things like leptin,
Right.
induced
saying, okay, that's enough, we have enough energy, we've taught ourselves enough. So have you looked into that in terms of like an outside source regulating breathing via that?
What, by the microbiome?
Yeah.
It's a very interesting hypothesis. We have not. That's well beyond anything that we've got to yet. That's a good point.
That's substantially, and now, another question is the reverse. A lot of essential amino acids like tryptophan, which is necessary for serotonin, also in the microbiome.
It's not for a restoration, but you can't remember the substance that it was, but released by the microbiome that drastically reduced blood pressure.
Is that right?
Yeah. I'm trying to think of what it was.
Well, my microbiome is screaming for pizza and beer. And I suggest that we convene down there, you can ask any questions there. Thanks, Tom, very much.
Well, thanks.
Actually, I wanted to give you a PowerPoint at some point, get some of those.
Well, it's on here now.
On there now, you have this in mind.
Or I can give you the,
Yeah, you can have it.
I've got, it's on a memory stick, so I can do it. But it's sitting right here. So, well, I wouldn't mind. Should we delete that? Sure. No, I don't want. That's tall. This one happened. This one's.
I'm sure you want to move to the second. All right, you have your stick, do you think you have that?
I do, yeah.
Thank you very much.
Appreciate it.
All right.